Hello Year 11. This is the very last lesson in the unit B2.4 Variation and Evolution booklet. And this lesson today starts on page 46 in your booklets, and we're going to be covering antibiotic resistance in bacteria, leading to the superbug MRSA, and warfarin resistance in rats. So two examples of natural selection in the modern day. And we're also going to be looking at something called the Human Genome Project, HUGO. So if you turn to page 46 in your books, and our objective, first of all, is to understand how antibiotic resistance has developed in bacteria. So in science, antibiotic resistance is when a bacteria that used to be killed by an antibiotic drug isn't affected by it anymore. In other words, it survives. You may have heard of a bacteria called MRSA, and it's often described as the superbug. MRSA stands for methicillin resistant Staphylococcus aureus, and that should be a lowercase a. Okay, so what does this actually mean? Well, this part here is the name of the bacteria that normally we have in the back of our throat. It's quite common at causing us sore throats, and because it's got a species and a genus and a species name, okay, we've got the genus is capital letter, the species is the lowercase a here. So what does this beginning part mean, the methicillin resistant? Well, methicillin is a type of antibiotic. And that means that this bacteria, Staphylococcus aureus, was first identified as being one of the types of bacteria that is resistant, does not get killed by this particular antibiotic. So it's not killed by the antibiotic methicillin. So sometimes this kind of MRSA infection, okay, can be cause issues like skin issues, okay, and sore throats and things like that, but sometimes it can be absolutely deadly, particularly in the elderly, vulnerable, the young, people who've got pre-existing conditions, okay. So we're going to be looking at making sure you understand what these words antibiotic and resistant mean and that you can use them confidently. Being able to explain how the bacteria have evolved using natural selection to become resistant to antibiotics. And the killer part is, could you suggest how we can stop antibiotic resistance? So you might have heard of this already, what doctors are suggesting we do to prevent antibiotic resistance. So, little animation, little cartoon. A natural population of bacteria has a variety, okay? In other words, there's variation. So if we look at this population of bacteria, we've got our green smiley face, we've got our yellow and green kind of like solemn face, and then we've got what I would describe as the monster bacteria, these star-shaped ones, okay? So in a population, there will always be some bacteria that are susceptible to the antibiotic. What that means is they get killed by it, and some that are resistant, okay? What that means is they don't get killed by it. So you might have a population of bacteria living in a body. So we start taking an antibiotic. So here's our population of bacteria, okay? We take an antibiotic, and when we take our antibiotic, the susceptible, weak bacteria die first. So you could see some of them disappeared, okay? Now, when we take an antibiotic, we're often prescribed it for at least 5 days, and we might have to take it 3 times a day or twice a day in order to kill off these bacteria. So we keep taking more, and more of the susceptible bacteria start to die. And that leaves us with a much lower population of bacteria, which is brilliant, okay? Because we now only have the resistant bacteria left. So you might think, well, the antibiotic's doing its job, it's getting rid of the majority of bacteria, and perhaps the immune system can fight these other ones that are left. But this is a problem, because these bacteria don't get killed by the antibiotic. and we've taken away all the other bacteria, their competitors. So now you stop taking the antibiotic, and what happens is bacteria reproduce. We know bacteria reproduce once every 20 minutes. 1 becomes 2, 2 become 4, 4 become 8, 8 becomes 16, and so on, until these resistant bacteria reproduce to very high numbers. Now there's lots of them. So you go back to the doctor and you say you're still unwell, okay, and you need to take the antibiotic again. So you take the antibiotic again, but this time none of the bacteria get killed by the antibiotic because all of the bacteria that are left are all resistant. They have outcompeted, okay, the other types of bacteria. If you like, because all the other non-resistant have been killed, these guys can have an absolute party and take over the body, okay? So you might have to take a different antibiotic to get rid of them. So what I want you to do is I want you to pause the video. On page 46, you've got some captions, A, B, C, D, E, F, G, and H. They're on the screen now for you to see. My advice would be to, first of all, decide what order these statements should go in. They're not currently in the correct order, so I would number them. And I'll give you a clue. If I give you the first one, you start taking an antibiotic is number 1. See if you can number the rest of them. Pause the video and have a go. So, our next one should be D. That's number 2. When you start taking an antibiotic, some bacteria are susceptible to and are killed by the antibiotic. Some are resistant or are not killed by the antibiotic. G is number 3. The susceptible or weak bacteria are killed first. In other words, the ones that are not resistant. Number 4, only resistant, strong bacteria are left. Number 5, you might be feeling better, so you stop taking the antibiotic. Always a good idea to finish the course that you've been prescribed by your GP. And E is number 6, the resistant strong bacteria reproduce. And number 7, the resistant strong bacteria are now more common. If you take the antibiotic again, it won't work because all the bacteria are resistant. So I want you to take these statements, and over the page on page 47, you're going to put these into the correct boxes. So you're going to write them in those little boxes there. And then I want you to draw some images, okay, to go in each of these to show me. If you don't want to be drawing fancy spiky bacteria with teeth, okay, you can draw bacteria that are triangles, squares, and circles, okay, and decide which one's going to be your resistant one and perhaps colour it in. So that might be my resistant bacteria, and you're going to start off with a whole variety of different bacteria. So keep it simple, your drawing. Pause the video and fill that in for me, please. So if you were following the drawings, okay, you may have used these images to help you, okay, or you may have done a simpler version of it. So A, you start taking the antibiotic. This is number 1. Number 2 here, some bacteria are susceptible to the antibiotic and some are resistant, so we've got variety. Our susceptible ones, we can see that this one's disappeared, for example, and this one has disappeared, okay, and this one over here. They are killed first. That's number 3, G. Only the resistant bacteria then start to be left, as these ones would start to die. You stop taking the antibiotic, and then these resistant bacteria start to divide and divide until they're more common. And then finally, if you take the antibiotic again, it won't work because all of the bacteria are resistant. So back on the previous page, at the bottom of the page, it says natural selection in bacteria is really rapid because they reproduce so quickly, about once every 20 minutes on average. So in the course of a day, bacteria can go through 72 generations, okay, of bacteria reproducing. So to avoid resistance building, to new antibiotics that are being designed all the time, doctors try not to prescribe them unless it's absolutely essential. And then they try to use a variety of different antibiotics. When antibiotics were first invented or discovered, we had penicillin. That was the only antibiotic that was available, and it was overused. And some people find that they have allergies to penicillin. It makes them sick, okay, it gives them a headache, that kind of thing. So your doctor, if you go to them and say, I've got a sore throat or something's wrong, they may say, I'll see if your body can fight it first before they start prescribing antibiotics. And they will never give you antibiotics for viral infections like flu or COVID, because viruses live inside the cells and they actually damage the cells and break them open when they reproduce. So in order to get an antibiotic to kill a virus, you would have to get the antibiotic to get into individual cells in the first place. Excuse me. So antibiotics are only ever given for bacterial infections. At the bottom of page 46, there's a little, very small paragraph with a couple of missing words. I'll go over that with you now. A similar problem has been encountered with some widely used pesticides. Pesticides are chemicals that kill insect pests that destroy crops. These pesticides, which have killed off most pests that are susceptible to it, leaving those that have the pesticide-resistant gene and are naturally resistant to it to survive, breed, and pass the gene on to the next generation. So you should be understanding that this is an example of natural selection. We have got variation up here, okay? We've got a mutation that causes some bacteria to become resistant to the antibiotic. This, for bacteria, it's not for us, but for bacteria is a survival advantage, okay? So that means that they survive here. And as a result, as they survive, they breed and they pass their resistant gene on to the next generation. So that VMA SPG, okay, applies here too. Now, when I wanted to show you this website about how rats have developed resistance to warfarin, I've used this for many, many years. And this year, it will not work because it's only supported by Adobe Flash. So I've had to be as inventive as I can. I'm afraid my animation is not great, okay? But I've tried to explain it with some pictures of rats. But it was, if you turn to page 48, it was these pictures that you can see of the little grey rat on the top is your regular rat, that if he eats the rat poison, he will die, and the rat poison is represented by the triangle, and the darker grey rat on the right hand side is our super rat, in other words our warfarin resistant rats. So I'm going to tell you a little bit more about warfarin resistance. So this is the poison warfarin, and you can see it's a rat and mouse killer poison. In bacon and cheese flavour with 2 ready-to-use bake bags, okay, that you can put down. Obviously, whenever you put any kind of poison down, you've got to make sure that it's not other animals like your pets that would be having access to this, particularly if it's like this lovely bacon and cheese flavour that's going to attract them, okay? So here's our rat, okay? And to begin with, most rats were killed by warfarin, okay? If they ate food that was poisoned with it, they would die. So our poor little rat here, Mr. Ratty, is dying. Whereas this rat here, okay, I've drawn him as slightly bigger, okay, just to show that he is our kind of super rat, but it doesn't necessarily mean that they are actually bigger. This rat has got a mutation that's happened to him that has made him resistant to warfarin. In other words, when he eats warfarin, it does not kill him, hence the nickname super rat. So, a little bit of information about warfarin. It was a well-known rat poison used on farms and in areas where rats were a problem, eating crops and causing damage to houses. It was used regularly, and the way it kills the rat is it causes them to often bleed to death. So it's not a very nice death for our rat. Warfarin is actually used in humans to treat blood clots and strokes, as it thins the blood and prevents blood clots. However, dosage in humans is always monitored closely. We don't want to be poisoning humans with warfarin. So this is a modern day example of evolution of the rat, as now warfarin really kills the modern rat found in our towns and countrysides. So there's absolutely no point in putting this stuff down in most areas because it's not going to be effective. So here's my start of my little animation. So originally all rats were pretty much the same, with no variation in terms of resistance to warfarin. In other words, okay, all rats, whenever they ate warfarin, they all died. So there we are. The crosses are indicating they're all dead. Then, due to a mutation in one rat's genes, this gave this particular rat the ability to be resistant to warfarin. In other words, it did not die when it ate it. So, this is our super rat. Here's our rat that is that's not resistant, and he dies, and this one is resistant to warfarin. The super or resistant rat bred and passed this gene for resistance to warfarin to other rats. And we know that rats and mice and rodents often have multiple babies in their litter, little mice and little rats, okay? And they reproduce very, very quickly. They have a very fast gestation period, so they can increase in their number very quickly. Not as quickly as bacteria, but fairly quickly. The super-resistant rats outcompeted the non-resistant variety until only resistant rats were left. So our little rat just disappears. So all we've got left now are these guys, okay, that are resistant to warfarin. So on page 48, I'm just going to move that back. It says, watch the animation. I agree, it's not the world's best animation, okay? And describe and explain, using the idea of natural selection, how rats have developed resistance to warfarin. Try and use that VMASBG, okay? But relate it to the rats for me, please. So this would be a 6-marker style question. Pause the video, have a go, and then I'm going to go over a mark scheme that will pretty much cover you for lots of different examples of natural selection. So, any kind of natural selection question that's a QWC, a 6-marker, is going to be looking for at least these 6 points. And you can see I've highlighted in black those words that we've been talking about all the time. Variation, mutation, advantage, survival, breeding, and genes, okay? So let's say you've got a population of giraffes or bacteria or insects or rats where there is variation. So in giraffes, it might be short or tall necks. In bacteria, resistant to antibiotics or not. In rats, resistant to warfarin or not. In insects, resistant to pesticides or not, or insecticides, okay? And for the second point, that these differences are all caused by mutations. Third point, it's an advantage if you're a giraffe to have a tall neck because you can reach more food, so you spend less time looking for food and more time breeding. It's an advantage if you're a bacteria to be resistant to antibiotics, as you don't get killed by them, so you survive. It's an advantage if you're a rat to be resistant to warfarin, the rat poison, because when you eat it, you don't get killed. It's an advantage if you're an insect to be resistant to insecticide, because again, you don't die when you eat the plant. Fourth marking point, as a result, more of the warfarin resistant rats, okay, or the antibiotic resistant bacteria, or the insecticide resistant insects, or the taller giraffes survive. Fifth marking point, these survivors with this particular advantage go on to breed. And final marking point, pass their genes for tallness, or for resistance to warfarin, or resistance to antibiotics, or resistance to insecticide, on to the next generation until the others are wiped out due to competition, or out-competed is a term that some people use. So check your answer and decide how many marks you think you've got, and I will be putting one of these types of questions onto Moodle as a rubric, as an extended response answer, okay, for you to have a go, submit to me, and I can mark. The very last page, page 49, is about something called the Human Genome Project, which I can remember happening when I was a pupil in school. There is a YouTube video link, and I will put this link, which is here, okay, onto Moodle as well for you to watch. There are 3 questions that I want you to answer. The video will tell you a little bit about what the Human Genome Project was, and the 2 different groups that were actually attempting to map the whole of the human genome. And then I want you to go on and write for me the benefits of the Human Genome Project, give some examples, and then what some of the issues, the problems are with having all of this data and this information. So what are the ethical, legal and social implications of the Human Genome Project, and give some examples. So this part of the lesson is all about understanding the potential importance for medicine of our increasing understanding of the human genome. And just to give you a little bit of insight, the human genome that was mapped was actually a combination of lots of different people's DNA. It wasn't just based on one person, but one person in particular who has contributed massively to the Human Genome Project is a lady called Henrietta Lacks. And Henrietta Lacks, if you want a little bit more information about her, you can Google her, okay, is in the news currently, okay, because her cells, she was a lady who had cancer. I think it was ovarian cancer or cervical cancer, and she went to the hospital and they collected some of her cells, but without her permission, okay, they grew up some of her cells. Now, Henrietta unfortunately died a few weeks later after they collected her cells, but they noticed that her cells never died, okay? Even though she died herself, these healer cells, which were the cancerous cells, they never seem to die. And they are what we call an immortal cell line. So immortal means live forever. And they actually used her healer cells, okay, to actually look at the Human Genome Project. And the reason why this is so in the news at the moment is because they've used Henrietta Lacks' immortal cell line cells to actually help them make the COVID vaccinations that are currently being given. So some of the information that scientists have gained from this Human Genome Project have been absolutely huge, okay? So I just wanted to explain that at the moment. It's very, very relevant, very current at the moment. So watch the video first, write your definition or your description of what the Human Genome Project is, and then I'm going to give you some information to help you with writing the benefits and the disadvantages. So, a couple of advantages of the Human Genome Project. It can help predict and prevent diseases by mapping the human genome, because we can know which genes will make people predisposed to particular conditions. That word predispose just means people who are more likely to have those conditions. So, for example, breast cancer, they know that breast cancer can sometimes follow in families. So if they can look at a person's genes and then look at a daughter's genes and the next generation's genes, they can predict, is that person likely to suffer from breast cancer at perhaps a later point in their life? And can we offer some kind of preventative course of action? So in the case of Angelina Jolie, she knew that her mother had had breast cancer and had died from it. So she had quite a drastic double mastectomy to remove both breasts to prevent any chance of her developing breast cancer later on in her life. Furthermore, Cures could be found for genetic diseases like cystic fibrosis that we've already talked about, and another disease, you might have heard of this, called sickle cell anemia. It's not the same as normal anemia that we sometimes see in teenagers or ladies after they've given birth. Sickle cell anemia is where the red blood cells turn into this literally like moon shape. And what happens, it means that they have less surface area to carry more oxygen, and they also get stuck as they're passing through blood vessels, and this causes a tremendous joint pain in people who suffer from this. Another advantage, improved medicines, okay? That personalised medicines can be developed based on the way our individual bodies react to the disease and the treatments, which will be more effective because these medicines will be tailored for our specific medical needs. Accurate diagnoses. Doctors can develop more accurate diagnostic techniques for certain conditions which are difficult to diagnose at an early stage. And it can also help us improve forensic science because genetic fingerprinting, okay, or profiling, as WJC prefer to call it, okay, helps us to match suspects to biological material found at crime scenes. And in the future, it could be possible to even figure out what a suspect looks like from their DNA found at a crime scene, i.e., what their eye colour, their hair colour and their skin colour might be based on their DNA fingerprint or profile, okay, that we have. Disadvantages, though, ethical, legal and social implications. Now, you would think, well, this is a piece of scientific research that was, in its day, it was amazing. I can remember them starting it when I was 16, okay, and by the time I got to university, they had completely mapped the human genome. So it was an amazing piece of research. So why would it have disadvantages? Well, one of the things that happens is that sometimes people don't want to know what potentially could happen in the future. So it can cause increased stress. People could be diagnosed with illnesses that they are susceptible to develop in the future. And then when they find that out, they might spend their life worrying about it even before they get it, okay, and not living the life that they should be doing, unaware that this disease is clouding their potential future. Some people would argue that and say, well, actually, they would prefer to know if a disease was going to impact on their lives later on, so that they could live their life in a different way. So different people have got different views on how they would approach this. So have a little think about how you would feel if a doctor could tell you exactly what potential diseases you might be more predisposed or likely to develop later on in your life. Would you change the way that you behaved, okay? And it does depend on what age, okay, you are told this as well. Then we've got something called genieism, okay, right? Genieism has got nothing to do with a genie in a bottle. It's people with genetic problems could be under pressure not to have children as a threat of passing on their faulty genes onto the next generation. So people, if they know that they've got something like cystic fibrosis, will often go and see a genetic counsellor. And a genetic counsellor will talk them through the risk factors, okay, of going ahead and starting a family and what the potential risks are and what they can do if a child is diagnosed with a particular condition, an inherited condition or not, okay? And this last one, this is your ethical and legal and kind of social disadvantages all rolled into one. Your parents, okay, will have life insurance, potentially house insurance, car insurance, okay, all different types of things like that. When they apply for a mortgage, they will have to state that they're healthy and well. If you go on holiday, you have to generally have holiday insurance, otherwise you won't get your money back if something happens on the holiday. This could lead to discrimination, though, by employers and insurers if they know, okay, if you have a higher likelihood of a serious disease, of getting a serious disease later on in life. So employers may discriminate against people who are genetically likely to get that disease and be favourable to those who are healthy. Insurers might charge people higher premiums, in other words, higher amounts to pay for their insurance, for holiday insurance, for car insurance, for life insurance, okay? And so do we want these companies knowing this information, okay? Or is this Freedom of Information Act, okay? This is your data, your genome, okay, that's being shared. So have a little think about how you would feel about that, whether you think it's a good thing or not a good thing, and the impact if insurers, I suppose, don't know this information before giving you a quote for your life insurance and your holiday insurance and car insurance and everything else. So just write some of those down, pause that screen, and we are at the end of this booklet. So I will be putting on over the half-term holidays an answer booklet for this particular unit. Apologies, the last answer booklet took so long. Because of all the genetics problems, you cannot write over with like this pen over publisher and documents or PDFs. So it was a little bit harder to try and put it all together for the Punnett square diagrams, okay? But there will be an answer booklet soon for this over the half term. Thank you very much, Year 11.